So yeah, thank you. Good morning. Thank you all for coming. I have what really is the great honor of introducing a student who has really left an indelible mark in my lab in multiple communities here at WashU and also at Harvard. And as I hope you'll see also in the field of neuroscience and machine learning, and that's Binshu Wang. So Binshu graduated with a bachelor's in physics from Peking University and has been a graduate student in DVBS since 2018, as well as a visiting fellow at Harvard Medical School since 2021. Binshu is a brilliant and creative thinker with a wide-ranging interest in the brain, extending from theoretical mathematical principles all the way to the electrophysiology, behavior, and perception. So Binshu got started in research in 2015, modeling human social behavior, population responses in macaque primary visual cortex using 2-photon imaging data, as well as developing algorithms for extraction of excitatory postsynaptic potentials in whole cell recordings. At every stage of their training, Binshu has been rightly recognized as an exceptional member of their community, receiving scholarships in physics, academic excellence awards, and the Excellence Award in Graduate in the City Award, which is the highest that Peking University grants. In 2021, Binshu also attended the MIT Brains, Minds, and Machines summer course, where they received the 2021 Fujitsu Laboratories Fellowship, and most recently, the 2022 Kwan Predoctoral Fellowship in Neurobiology at Harvard. So Binshu has also been a remarkably productive scientist. In the past 5 years of graduate training, Binshu has published five to six peer-reviewed articles in competitive machine learning conferences and general interest journals, including Neural Information Processing Systems Conference, NeurIPS, the International Conference on Learning Representations, Gecko, Nature Communications, Cell Reports, and another manuscript coming in this year. So what you're going to see today is just a fraction of what Binshu has achieved. And I was fascinated to see it all happen from the very beginning. I first met Binshu in August 20th, 2018 at the orientation faculty poster session at the Ferro, just next to this building. It was a day that I remember being very exciting for all the different new beginnings. Obviously, the beginning of Binshu starting their PhD, I was starting my lab. And Binshu was among the first to approach my poster. And I remember distinctly that, after asking some typically brilliant, insightful questions, I just leaned forward like 3 times and I was like, tell me your name again. Tell me your name again. Because I knew that this person was going to be very important in my life. And perhaps as somebody sitting in the audience asking me all the questions that I hadn't thought about during a talk, or somebody that I could approach with specific questions to bounce off ideas. But instead, I got lucky and I got somebody that eventually joined my lab. Binshu, as I mentioned, has had an interest in vision and machine learning from a long time. So when they were interested in rotating in my lab, I wasn't surprised. At the time, though, we had just launched a new computational system. And this system worked. It was something that basically linked a novel class of deep learning models called generative networks, which are made to synthesize images. And we had linked it with electrophysiology, neurons in visual cortex of the monkeys. And this computational system worked pretty well, well enough. But given that it relied on deep networks, it still remained a little opaque. We didn't quite understand how the whole thing worked. But it worked. So I just wanted to go ahead and start moving and using it to start gathering experiments and get some data. But when Binshu started in the lab, just with a rotation, the more that they read about this and learned about the system, the more that Binshu commented, there's no way this can work. And part of the reason is that this is a system that works by optimizing images using a generator that deals with 4,000 dimensions. And we made it work by just taking a few samples, a few dozen samples at a time. And that's crazy. That would be like trying to go to Kansas by taking some random steps in the room with a noisy compass. It really shouldn't work. So basically, Binshu wanted to explain this and wanted to delve into the process of how the whole thing works. And at first, I felt a little reluctant because although I did welcome the exploration of this method, I knew that it's a common mistake for new labs to not to just kind of spend too much time on technology development and not really getting to the brain right away. So what I proposed was, Binshu, why don't we just start doing experiments, and then we can, and after that, do some computational explorations. And Binshu basically said, I can do both. And Binshu was not lying. Binshu works really well at night. And so when I would come in the morning to start to set up animal training, Binshu would come with all the simulations already performed, basically illustrating how to optimize the whole system, how to make things faster, and exploring ultimately how it worked. And so what Binshu found was that the reason that the whole system worked was that the generator, although in name had 4,000 input dimensions, actually most of those dimensions were dead. Most of those dimensions were not coding for images. So the whole reason that the system worked was that it was inefficient. And so Binshu had found that neurons and the optimizing searching algorithm had just found a manifold, a submanifold in this larger space and explored it. And so that's why the whole thing worked. And with this kind of investigation, which became Binshu's first publication, we basically gave us like a solid steel foundation for the rest of our research program, made everything go faster. And we collected hundreds of experiments over the next few months because of this improvement in performance. And I'll note that in addition to this great computational work and the physiology experiments that followed, there were hundreds of them, and Binshu did half of those experiments. I mean, and I'll say that that's a big achievement, because monkeys really are, they're very smart and they're very stubborn. And to work with them, it takes a certain intuition and ability to know when to stop, when to nudge them, how to really push everything forward and get the best collaboration with them. And Binshu was amazing with these monkeys, especially with one monkey named Beto. This is a monkey that the experiments that these two did together became some of the landmark experiments in every paper that we have published. And so recently I had a very rare opportunity to retire a monkey to a sanctuary here in the Midwest. I had to pick, I could only do one. And I always thought of these two working together, Beto and Binshu. And so I knew that it had to be Beto. So now Beto is out here in Indiana, living his best life, hopefully with a pool, with the weather these days. But yes, so today I'm here to prepare to let go of another beloved primate in the lab, Binshu. And as Binshu now prepares to defend this thesis, I do feel a mix of emotions. I feel a sense of loss at the fact that Binshu is departing, but I'm also exhilarated by what the future holds for Binshu, for the field of neuroscience and machine learning. And now as Binshu joins the brand new Kempner Institute for the study of natural and artificial intelligence in Boston, I'm pretty sure that this is a person that is also going to be shaping our futures as well. So ladies and gentlemen, join me in welcoming an individual whose intellect really has inspired me like no other. So Binshu Wang.
Thank you for so kind introduction. Can dim the light a little bit to show the screen? So today's my thesis defense on the topic of charting the landscape of ventral stream neural tunings on the generative image manifolds. Oh, here. Hello. Cool. Now I can hear you. So yeah, today my topic is to chart the landscapes of ventral stream neural tuning on the generative image manifolds. So first, let's take a moment to close our eyes and imagine you're a neuron. So our brain lives in a cavity filled with fluid contained in the black box. And our minds build up the 3D space, where it creates an illusion that there are objects occupying the space and we can interact with. So the neurons in the brain, they didn't sense light, not in the normal conditions, and they didn't see things. So it only relies on the representations of other neurons to get to know to the world. So they didn't see the scene here, but they see the spikes. So in this sense, the sensory system holds a gateway to the whole brain and the higher cognitive functions. So I think study the neural representations of the sensory informations is the foundations for understanding all the higher order functions of the brain. Moreover, in the age of the deep learning and artificial intelligence, understanding the representation has another meaning, which is we can align the representation in the brain and the machines. So if we can achieve aligned representation between the two, then perhaps we can decode the visual imagery from the brains and more control the brains, or even building more robust computer vision systems in the machine. So this motivates my studies of the sensory system in the visual cortexes. So first, I want to ask the meta level question. So how should we study vision? So let's get back to the research that gives the Nobel Prize in 60 years ago, which is the human visual studies of the complex cells in the simple cell and complex cells in the cat visual cortex. This is a video excerpt of their study. You can see that they are showing different types of stimuli, the light bars, different orientations and positions. Showing these different parameters, they're trying to find which position and which combination of these parameters, and elicit the highest firing rate of the neuron. Then do this by hand and find the optimal locations and also the orientation. We think this marks the search for optimal stimuli in visual neuroscience. To take it on a higher level, the paradigm for visual cortex studies is usually like this. So we tend to choose a set of images, I, and we record neural responses to these images using like either electrophysiology or imaging. And then we infer the neural function based on the pairs of responses and the images. Usually, we assume that the high response images are more interesting to the neuron. The problem for this study is that the natural image space is immense. So assume you have a 64 pixel RGB images. It already has 12,000 dimensions. If you assume there are 2056 possible values for each pixels, then you get this immense number of combinations of values. This is totally intractable. So how should we choose our stimuli for visual neuroscience study? Here we look back into the history of neuroscience again. So fast forward from the human research, we have known so much about the visual systems of primates. So the visual information comes from the retina towards the LGN in the thalamus and going through a series of cortical areas in the visual cortical regions. And we are specifically interested in the ventral stream pathway, which are in charge of the object recognition and shape perceptions. So for each cortical regions, people have developed a series of shape spaces that are preferably used for this space. For example, in V1, people tend to use the oriented bars or gratings. For V2s, the shapes formed by the line segments. For V4, the curvatures and contours or the non-Euclidean gratings. And in even higher level spaces like the IT cortex, people find the hands or faces are useful stimuli for them. So among these approaches, there are two types of strategies. For lower level visions, people tend to choose very simple but parametric image spaces. For example, from V1 to IT, people might choose to V4, people might choose these orientations like textures or this shape stimuli, and they live in a low dimensional parametric space. But for IT, people tend to choose another strategy, which is to use the potentially isologically relevant stimuli like faces, objects, or just natural scenery or images. these two approaches has their own pros and cons and costs and benefits. For a low-level approach, you enjoy the parametric space, which allows you to do dense sampling and draws smooth tuning curves like that you see in the orientation space. But on the other hand, it suffers from low diversity. So maybe your space doesn't contain the preferred stimuli of the neuron. For a high-level vision space, you really use this natural image samples. It's non-parametric and it just allows sparse sampling, but it has a high diversities. And in that case, normally you see the results as a categorical selectivity to some image categories or response barcode. So we have one visual systems. So can we unify these two strategies into one single approach? So let's remind ourselves that the natural image themselves has certain structure. So look at these cute cats, they look cute. And also they are natural images. So if you shuffle their structure statistically and disturb certain statistical structure, it doesn't look cute again. So this means that natural images have to obey certain statistical laws. And by constraining these statistical laws, they're living on a sub-manifolds of the whole image space, which has much lower dimensionality than the whole pixel space. And on this manifold, if you traverse it by traveling on this manifold, the images will continuously morphing into each other, but state looks natural. And we estimate it's like 100 to 1,000 dimensions natural image manifolds. This idea per se is still certain abstract, but the recent advances in deep learning and generative image modeling makes this idea much more concrete. So generative adversarial network is one type of generative image models, and it maps the vectors from a latent space to an image space. Further, if you continuously morph the latent vector, the images will continuously transform into each other's. So geometrically, this is like a smooth parameterization of a manifold from the latent space to the image space. So previous research by Carlos has found that the GANs has encompasses diverse image sets. So if we pick some classic images in the top row, like the shapes, curvatures, 3D shapes, or even object images, you can find the corresponding images in the GAN image space, which means that the manifolds parameterized by this generator encompasses all the previous classic image spaces. So this shows that the GAN is a good candidate or surrogate for the natural image manifold. And we are going to study the visual coding on it. What are the benefits of this approach? So we think it unifies the pros of the two classic approaches. First of all, it's parametric. So it allows dense sampling and smooth manipulations of the images. And also it allows local search, which I will introduce in a second. And also it has high diversity, which means that it can express complex features that could be preferred by the high-level neurons. So we're going to study the visual tunings on this manifold. So how do we understand this tuning? If we assume certain regularities or continuities of the neuron tunings on certain space, then we can think about this neuron tuning functions like a landscape. So on this landscape, the x and y axis on the horizontal dimensions are the image axis or parameters to the image space. On the z-axis, or the height of the mountains, are the neuronal responses. So the peaks on this picture are the highest response the neurons can reach, and also corresponding image. So we call this picture the tuning landscapes. And my thesis is devoted to study this landscape in different approaches. We split it into several parts and asking different questions. First, we want to ask where are the peaks located on this manifold? And then we want to measure what's the shape of the peaks and their global geometry with each other. And finally, we want to know, does the tuning landscape of the neuron differs on different image manifolds? So let's get started. Our first project is to search for tuning peaks. Our strategy is just like what you do in the hiking. You start from somewhere and then just keep climbing until you reach the plateau and cannot go higher. So mathematically or geometrically, it's like the following the gradients of the tunings and then gradually morph or change your images until you reach the plateau. To be fair, this approach is not that novel in some sense. Like even in the classic vision neuroscience research, people are manually doing that. As you've seen in the Huber and Wiesel's video, they're manually changing the parameters of the orientation and positions and optimize that parameters. And people have also tried to optimize the orientations, spatial frequency, and other parameter of the classic image space. In 2001, people have tried to use gradient ascent on the pixels to synthesize the patterns that optimally stimulate V1 neurons. And you can already see this preferred patterns of the V1 neuron can be synthesized from scratch in the pixel space. Starting from 2008, in the corner lab, they have been searching for optimal stimuli in the shape space of 3D shapes. And using an evolutionary approach, they can reach highly activating shapes for IT neurons. So in our approach, we are taking these evolutionary algorithms to the deep generator space, which is developed by Carlos, Will Shell, March Livingston, and Gabriel Kleiman. So in this approach, we start with a bunch of latent codes or random locations in the manifold, and then we turn them into the generator space, into the images. And then we show the images on the screen. And the monkey is sitting there looking at the screen while we're recording the spikes from the monkey's brain. And the spikes are summarized as a scores, like the mean firing rate in a certain windows. And the scores are feedback into optimizers. And the optimizers are going to propose the next batch of latent codes. So by iterating this approach, like tens to hundreds times, you're going to morph your images and reach a high firing rates image. So that's where I was when I started my PhD. And as Carlos had mentioned, I find this approach a little bit puzzling. One thing is, it shouldn't be able to optimize so many parameters. So I did some computational research on the geometry of the GAN image space and find that it has highly anisotropic structures in it. For example, if you travel the same distance in the latent space of some GANs, like at some directions, you're changing this dog to close, but in some other directions, the dog doesn't change at all. So there are certain non-space in the GAN image manifolds and a certain anisotropic. So without knowing this, we may be not able to optimize very efficiently. Further, I noticed in the generator we use, there's a certain spherical geometry in it, which means that if you travel on the radial axis, you are majorly changing your image contrast, while you are changing on the spherical dimensions, you are changing your image patterns. And we are more sensitive to the spherical changes of the images. So those are the more meaningful perturbations to the image sets. And also we leverage this geometric observations and benchmark and develop a new efficient search algorithms based on the Cholesky CMES. And it navigates the 4,000 dimension space much more efficient than the previous counterpart, the genetic algorithms. And with the same fixed number of images, this increased the achieved firing rates by like 44%, and it saves a lot of experimental time. So leveraging this technical improvement, they collected a large sets of optimal images or highly activating stimuli we call prototypes for cortical regions like V1, V4, and IT. So in summary, in this first part of the talk, we did some technical improvements such that we can climb the tuning peaks of the neurons efficiently. And then we can find the prototypes or highly activating images. Next, I want to ask, what is the shape of the tuning peaks or the tuning landscape in total? What we did is also like what we do in hiking. So you usually first climb to a peak, and then you look around to see the scenery. The only challenge is you have 4,000 dimensions. So this is like the elephant in the room. How do you study the shape of tunings in the 1,000 more dimension space? So we designed this following manifold experiment to help us explore around the peak. We find that when you explore in the latent space, your trajectory usually look like this sinusoidal structures with some mass behind it. And the PC1 directions of this trajectory is usually the peak dimensions, or pointing close to where you are at the end of the evolution. And by doing PCA of these trajectories and finding the PC2 and PC3 and other PCs, you are able to create this reference frame for the neuron, and then create this low dimensional submanifold in the high dimensional space. On this 2D sphere, each of the locations is one image. And then we sample the azimuth or elevation grade on this hemisphere. And each of the grade points is a stimuli we use for a neuron. And if you travel continuously on the manifolds, the image is also changing continuously. So during experiments, we already first localize the receptive field of the neuron by selecting a unit based on the receptive field and PSDH. And then we conduct the evolution experiments, which climb the peak and search for the optimum. Then we conduct the manifold experiment, which explore around the peak and chart this two-dimensional submanifolds in the image space. And finally, we present some reference images to the neurons and measure selectivity to other classic image spaces. During the experiments, the monkey is sitting there like fixating on the screens. And we follow the RSVP paradigm where the image onset is 100 milliseconds and offset is 150 milliseconds. We have 3 to 5 images in the block. We have data collected from 2 monkeys, A and B, and we embedded 64 channels electrode arrays in the 3 visual areas, V1, V4, and IT. And we have 45 sessions for each of the monkeys. So let's look at the results. This is one illustration of the evolution experiment. So during the experiment, you see the image is continuously changing from a gray colorless patterns to reach something that may look like an ear or hand. And the firing rate of the neuron increased by nearly 2-fold. And down there, you see the peristimuli time histogram or the response dynamics across the process. So then comes the manifold experiment. We take these images resulted from the evolution experiment and morph it on the 2D grid like this. So each of the grid points is visualized as the image here. And present each of them to the monkey and record neural responses to each of it. Averaging the neural responses illustrate this beautiful 2D tuning maps around the peak, centered around this image. We also tried other subspaces by perturbing on the other dimensions like PC45 or PC50, which result in different image grades and different submanifolds. But we also see a bell-shaped tuning map. Even when you traverse random directions using 2 random vectors, you see a bell-shaped tuning maps. So we can conclude that the tuning maps around the prototypes are generally smooth and bell-shaped, which is kind of expected when you optimize to a certain peak. or the optimum. And this is reminded of the classic radio basis function, the machine learning literature. We collected these tuning maps for the channels in V1, V4, and IT, resulting in this montage. And you can see they generally follow this bell-shaped structure with some certain variation across the areas. We quantify them using the kind function visualized as here. It's an analog to the 2D Gaussian, and but it's defined on the sphere. So it contains a center parameter controlling where the peak is, and it has a sharpness parameter kappa, which controls like the sharpness of the peak. If it goes up, you see the peaks becomes sharper and narrower. This is an anisotropic parameter, the beta, and if you go up, the peak will become more anisotropic and even becomes two peaks when the parameters exceed a certain ratio. So it fits this Gaussian tuning, the Kent tuning functions for neurons. And we find that generally it fits well over 0.7 R squared. And the distribution of fitting parameters is as following. So the center of the Kent function, or the peak location, is located generally in the center as we expected, but it's slightly biased towards the PC3 direction predicted by the evolution experiments. And also the shape parameters are mostly unimodal, bell-shaped instead of two peaks. So we conclude that the tuning maps are well fit by Kent function, and generally it's a unimodal sitting on the center. Next, as we motivate us by comparing us to the classic image spaces, how does the tuning in the generator space compare to the classic image spaces? So we developed this radio tuning curve analysis, which generalized from the classic tuning curves. Firstly, you find the peaks in a certain sets of images, and then measure neural responses as a function or the image distance to the peak. And we use the image similarity metric, LPIPS, a convolutional neural network-based metric to compare the image similarities. So we find that in this plot, the y-axis is the response, and x is the distance to the peak image. So for example, in natural image sets, the peak image is a monkey face. And by deviating from it, you see the faces of a distorted face, and also other faces in non-optimal configurations. And in the GAN space, you see the peak image also looks quite like a monkey face, but it charts, when you move away from it, it smoothly charts its tuning curves. In some other image space, like the curvature space, it seems that it's not locating on the peaks of the neuron, so it doesn't elicit a high fire rate, and doesn't has a high range of the diversity. So we quantify two aspects of this curve. One is the peak activation, or how high the mountain is, and the other is the width, or how wide the mountain is. So comparing the multiple spaces, we find that the GAN image space elicits the highest peak activations, which is seem to be a result of the optimization process. And by comparing the width of the tuning with other spaces, we find the tuning of the GAN space is also wider than the classic ones like Gabor's or curvature space. But it's slightly lower than the natural image space, which we can discuss in the future. So we even compared V1 neurons, the manifold image space, and the Gabor's, which is the classic image space for V1 neurons. We saw that the manifold image space can elicit a higher peak activations, and also charts a wider tuning curves, which means that the peaks of this V1 neuron can reside slightly off the Gabor's filters space. So in summary, we find these tuning maps in a general image manifolds, and they have spans a larger dynamic range or higher peaks ratio than the classic image spaces, and also it spans a wider image domains than classic ones, which is closer to initial images. Finally, we want to understand how the tuning. So there are certain type of property of the landscape. Some properties are local, like the peak and the tunings around the peak. We use the manifold experiment to characterize this. And there are certain global parameter properties, like the prevalence of the peaks, or we can use the evolution trajectories to chart this global geometry because it traverses the whole landscape. So we compared these landscape parameters for the CNN hierarchy and the ventral stream, because people have found that the pre-trained CNN models are generally relatively good models for ventral hierarchy. First, we found that the tuning width around the peaks sharpens along the hierarchy in vivo. When we look at the tuning maps for V1, V4, and IT, you see the peak sharpness around the prototypes becomes sharper and it falls off faster. We can characterize this by the kappa parameter of the Kent fitting or some non-parametric test. And they all yield consistent results that IT has a sharper and narrower peaks than that of V1 and V2. We found highly similar results for the in silico visual hierarchy in the CNNs. So the first layer have a much broader tuning peaks, and the last layer has a much narrower one. And we can reproduce the same analysis, showing that the sharpness grows as a function of depth. As for global properties, the search trajectory reflects certain part of it. And we first characterize the search convergence time. We found that in the in vivo visual hierarchy, the search convergence time increased as a function of the layer. In V1, it converged in like 12 iterations, but in IT, it converged much slower in like 30 iterations. And we characterize the same properties for the in silico visual hierarchy, the CNNs. And we find that also the first layer converged much faster than the last layer. And the last layer may not have converged in this time span. So this shows an increase of convergence time for search, which shows that the peaks may be sparser or harder to find. We also designed a reduced dimension evolution experiments. In this experiment, we search in two spaces. One is a 40,000 dimension full space. The other is a random 50 dimension subspace. The geometry idea is the following. So if you pick a random 50 dimension subspace, you're kind of picking the slice in the latent space. And it may or may not intersect with the peak of the neuron. If the peaks are very prevalent, then this intersection will be often, and then you can optimize well. If it doesn't, then it will not reach a peak in any sense. So we recorded this for V1 to IT and find the detrimental effects of the dimension reduction increase along the hierarchical level. In V1, it's very surprising to us that even you are optimizing in a random 50 dimension subspace, you can have a very small gap between, or no gaps between the 50 dimension subspace and the full space, showing that the peak is so prevalent. But in V4 and IT, you see the gaps gradually increase, showing that the peaks become less prevalent and harder to be intersecting with this subspace. We quantify the activation ratio between the two threads and show that a decreasing trend from V1 to IT. We also redo this for in silico visual hierarchy and find that also an increasing gap between V1, conv1 or the first layer to last layer. And this grows as a function of the depth in the hierarchy. So in this section, we have observed 3 consistent progressions in both the ventral stream and the convolution neural network models of the ventral stream. So we find the tuning peaks grows, the width of it decrease along the hierarchy, and the time to reach a peak increase. And finally, the required search dimensionality increase. So these are all very interesting observations about the tuning landscapes. Finally, we want to understand what is the underlying geometries that produces these trends. So here, we just use a very simple model to illustrate why this could be possible. We build a toy model of the neuron tuning, which is a Gaussian bump in the 4,000 dimension space. And this bump has certain proper parameters. It has a center parameter Z0, which defines where the peak locates. And it has the sigma parameter, which is the tuning width along the tuned dimensions. And there's a D parameter, which controls how many tuned dimensions there are. So this part, we want to change this parameter of the tuning bump and see what kind of combinations would reproduce the trends we see in vivo. So this is an intuitive 2D examples with the corresponding sigma and the D's here. So in the first part, you see the sigma is like low, and there's one tuned dimension, which is the horizontal dimension, and there's an untuned or invariant direction, which is the vertical dimensions. In this case, if you run a 1D subspace in the space, you are going to intersect with this tuning bump very easily, nearly always. But if you have a 2D tuning bump like the lower ones, you're not going to run into it very easily. So as the tuned dimension number decreases, it's easier for a random subspace to intersect. And also, if you increase the tuning's width, then the neurons are going to evolve faster because it's easier to find the gradient and to climb the peak. If you are mathematically minded, the intuition of this is the Gordon escape theorems, which means that dimensionality of random subspace to intersect this certain subset is when it's higher than a threshold, then it's nearly guaranteed to intersect. And this threshold is corresponding to the square of the Gaussian width of the sets. So we did this analysis or numerical experiments for 3 of the experiments we have done, the evolution speed, manifolds, and the reduced dimension evolutions, and plot the corresponding statistic on the 2D phase diagrams of the tuning width and also the tuned dimension number. We find that if the neurons follows a increase of tuned dimension number and a decrease of tuning width, we can qualitatively reproduce the trends we saw in vivo, which means the search convergence time increase along this trend, the tuning width decrease, and also the gap of reduced dimensionality increase. So in summary, we find that evolutions can find the local optimums on this landscape. And with proper sampling of the stimuli, the high-level neurons can also have smooth bell-shaped tuning curves in this manifold. And the ramp-shaped and bell-shaped tuning could be 2 views of the same mountain, depends on how you reach there. And finally, the tuning landscape of neuron follows the progression along the visual hierarchy. The higher visual regions exhibit sharper tuning peaks, and those will have more tuning axes. And this concludes the second part of the talk. So in the previous 2 projects, we are all using a single image manifold, which is our generator. So even it's very expressive and comprehending, it has certain limits. And for example, in the schematics, if the peaks locates outside this manifold, then it's hard to reach there. So we want to try, if we have multiple image manifolds, can we study the difference of tuning landscape in multiple of them? And does the neuron aligns better with certain image manifolds? So in this part, we studied alignment and neuron tuning with the parameterizations of pattern and object-based image manifolds. So let's forget all about the math and think about 1 stories about 1 patrons and 2 painters. So imagine you have a patron who want to buy a painting, and he ordered it from 2 painters of different styles. One is a classic image artist, and the other is a modern artist. And they are all trying their best to produce the best drawings for the patrons. and patrons providing feedback to these painters for them to improve their paintings. But in some sense, some artists will have an easier time to produce the preferred art of these patrons. Like the patron just intrinsically like the modern art, then the classic artist is hard to produce the favored painting for them. So this story is very similar to our case for the generative image manifolds. Our generative manifolds are GANs, are our painters. And they have different styles. And the neurons, in our case, or the patrons, are different functions in the image space. And so if the tuning functions and the GAN image manifold aligns in a certain fashion, we will have an easier time of optimizing or searching for peaks on that manifold. So do neurons align with some manifold better? First of all, let's get to know our painters, the two of them, like reminiscent of Carlos. And one of them is the DeepSim, which has a single image space, 4,000 dimension latent space. And the other is a BigGANs provided by DeepMind in 2018. It has a factorized latent space of 128 dimension subspace and for class and for noise. So they have different neural network structures and different depths and different many things. And you can see the image qualities or statistic differs between them. The DeepSim controls the patterns on the canvas without respecting to the object. In this illustration, you see the center image look like a vessel, but if you traverse the image manifold of the DeepSim GANs, it can turn into like a peach-like object or like anything like a door or something. So it's basically morphing the patterns on this image canvas. But instead, in the BigGANs, if you traverse in the object identity subspace, you see the same animal will change into other animals like dogs, cats, or maybe like rats. And if you traverse the object nuisance transformation space, this vessel will transform their positions and location and pose in this canvas, but they're still the same identity or same object categories. We also find that the GANs have very different image statistics. We use a common image distance metric called Fréchet inception distance to measure the difference of our two image distributions. We find that the BigGANs has very close image statistic to the ImageNet, which is the commonly used object database. And the DeepSim GANs has a much higher distance to this natural image space, but it's still lower than random noise or pink noise. To summarize, this BigGAN has some object-based parameterizations and has a compact latent space, just 256 dimensions. And the image statistic is much closer to ImageNet and photorealistic. For DeepSim, it's pattern-based parameterizations and has a much higher dimension latent space, 4,000 dimensions. So it's highly expressive, but the image statistics is more different from the ImageNet, and it's more textury and has lower objectness. So here's our experiments for this part. We generalized our evolution experiment to two threads, parallel evolution experiments. We have two image generators like this. One is the DeepSim, one is the BigGANs. And we have developed two optimizers corresponding to each one of them. Both of them are CMAES, but with different parameters. And in each generation, the two generators will create their own images, which are merged and interleaved as a string and show on the screen as a rapid serial visual representation experiment. And the neuronal firing rates are also recorded, and the firing rates are sorted into two streams to feedback into the two evolution experiments. So let's see this one in action. So in the first generations, they start from different things. In the DeepSim, we start from this texture, colorless texture. And in BigGAN, it originally start with an object. as the evolution going, you see the firing rate goes up in both spaces, while they create highly different image patterns. One is this dark browning gradient, one is this egret. But they are reaching similar level of firing rates of the neuron. So, and this difference is not statistically significant. So this at least shows us the neurons can guide evolution in very diverse image manifolds and reach similar level peaks. And in the neuron's eyes, these two things may be highly similar, maybe because they share some embedded motifs in it. So the direct question we want to ask is which of the artists capture the favor of the ventral stream better? So we first quantify the success rate of each of these generators. We use this analysis, which by comparing the firing rates of the initial two blocks and the two blocks that evoke the highest firing rate, and do the two sample t-test between the firing rates in these two collections. And we find that the success rate of the two image generators differs in their trends. We find that the deep sim image space has a near 100% success rate for both V1 and V4, and it has like around 60% success rate for IT cortex, which has a decreasing trend along the hierarchy. On the other hand, the big N nearly never succeed for V1 neurons, and it increased to 50% for V4 neurons and to 60% for IT neurons. And this trend is nearly opposite to that of the deep sim. So what's the intuition behind this? So imagine you are a V1 neuron, and the big N painter tell you a cat, and you happen to like it because the cat has stripes. And the stripe just aligned accidentally with your preference of orientations. And then the optimizer says, okay, since you like cats, I will give you more cats. And it perturb in the object image space and give you more cats of different orientations, different colors, different poses. They're all cats, but they lack the feature you like the most, which is the patterns on the head. So in this case, V1 neuron is confused, and it cannot guide the evolution anymore. So this is our current interpretation and understanding of why the V1 cannot guide the big N's evolution so well. And so it seems that the tuning of the higher visual regions are increasingly aligned with the big N's. We also compare the firing rates of the neuron. Before optimizations, the big N or the red dots have higher firing rates than the blue dots, the deep sim. But after optimizations, the trend is flipped in V1 and V4s, which means even the big N start higher, the deep sim can climb the higher peaks than those in V1 and V4s. But in IT, the two optimizations reach comparable firing rates in the two spaces. So as an intermediate summary, we find it's easier to climb the tuning peaks of the V1 and V4 neurons in the deep sim space than the big N image manifolds. And it's comparably easy or hard to climb the IT neurons on the big N image manifolds. So after finding this interesting trends, we want to look deeper into this trends of IT's preference of objects. So in the visual neuroscience literature, there has been a long history of the neurons dynamic encoding of object images. So this is one figures from the Van Nassen's paper in 2006 or something. And they found that in the earlier neural responses, it responds to near everything in the object space. And then in the later responses, the response becomes more specific to a certain subset of it. This shows that there's a coarse to fine, or broad to sharp tunings progress in time. And also others have found that the neurons tuning start from linear subunit tuning to nonlinear subunit tuning. And also population response start from a more correlated response to become more decorrelated and high dimensional. We have observed similar things in our previous projects of the manifold. And we find that, so in the upper part, we showed this image grid, which are our tuning manifold, as you see in the part 2 of the talk. And we are going to show this on the grid of responses, which correspond to the electrode locations on the cortical area. So if you visualize this map on the cortical region, you see that different electrode or different neuron sites have different similar but different tuning peaks on this manifold. But this is a static view of this data. If you add dynamics to it, which means average firing rate of different time windows, you see something different. So let's wait. So you see at the start, it has no responses, but after the initial response, the neurons, nearly all the landscape lights up. And then the neurons differs and into different individual tuning peaks and becomes more decorrelated. So we want to explore similar ideas in the two manifolds, the deep sim and big GANs. Before diving in, I want to say that there are two sense of time or two sense of dynamics. In one sense, it is the evolution progress, which is the units of blocks. So from top to bottom, you see the progression from the first block to the last block. And on the x-axis, it's a progression of time within each image presentations. And this is a unit of milliseconds, from 0 to 200 milliseconds. And this is the data we are analyzing, which are these two blocks of the time of evolution by time of the dynamics. So you can qualitatively see that in the evolution in two spaces, the deep sim and big GANs have slightly different dynamics in the response and evolution-wise. We plotted the average PSDH for the three regions evolutions. And before optimization, you see the big GAN PSDH is slightly higher than the deep sim, as the average has shown. And after optimization, also like the average showed in V1 and V4, you see the consistent increase of firing rate in all time bins exceeding that of the big GANs. But in IT regions, you see a switch in time. So in the earlier time bins from 50 to 100 something milliseconds, the deep sim seem to be preferred for evoking a higher firing rate, and the big GANs seem to have a higher firing rate in the later time bins. So this motivates our analysis of which part of the activation does the neuron recruit. So let's remind ourselves that in the evolution experiments, the objective window is 50 to 200 milliseconds, so which is this whole part of the firing rate response. But it's a liberty of the neuron to decide which part of the firing rate to increase in this time bin. So it could decide to increase the transient response or the initial response, or it could increase the later and more sustained response, depending on the preference of the neuron. So we take a difference between the max response and the initial response, and then attribute it to different time bins on the time axis, which result in this green curve and this yellow curve. So we compared this attribution between the two spaces and see their difference. We saw that for V4 space, the deep sim and big GANs increase the firing rate in a comparable fashion across all the time bins. But for ITs, the big GAN attributes recruit more responses in 100 to 120 milliseconds. And in contrast, deep sim recruits more in 50 to 70 for the transient response period. This shows that they have different dynamic preference to the two image spaces. We also compared the optimization dynamics in each time bins. So we take the PSDH in each block, average PSDH of each blocks, and take the firing rate in different time windows in this PSDH. So in the initial time windows, it will be flat, and then you will see the transient response evolutions, later response, and the sustained response. We saw that in V4 space, the blue trace or the deep sim consistently exceeds the big GAN uniformly across time bins. But in contrast, in IT areas, in the initial time bins of 50 to 100 milliseconds, the deep sim still evolves faster and reach a higher firing rate. In the later time bins, the higher firing rate is achieved by the beacons, and it's nearly always stayed on top. This also illustrates the same points that in V4, either early or later response preferred or more aligned to DeepSim or the pattern image manifolds. But the later response of IT is more aligned with beacon, which is source of their object preference. Finally, want to investigate how the neuron-guided evolutions change the images. And we're specifically interested in one aspect, which is the objectness of the images, which is how object-like is an image. And as you know, object is kind of a perception of us, so it's not well-defined and depends on the perceptual system. So we use an object detection system called YOLO as our surrogate for the perception system. It gives us a detecting box when it sees an object, and the confidence scores for how confident there are objects in this detecting box. As you can imagine, there are variations of objectness in each image space. For example, this bird is detecting with 93% confidence, which means like we can see it very clearly on the background and it's clearly an object. And some other things like the sceneries, like this stock or like a random object, is harder to detect and be classified as object. In the DeepSim space, there are also varying degrees of objectness. Some images, like this one, is decoded as a teddy bear with a very low confidence. Perhaps it's the texture of it seems like a teddy bear. And here, it looks like a dog or a black dog. Maybe it sees like some black dog very often. But on the distribution level, the beacon's image space has a highly similar distribution of objectness or confidence scores as the ImageNet per se, and they are highly aligned. And for DeepSim, it has a lower detection rate, which means the model sees a smaller fractions of objects in it. And also when it detects, it has a smaller confidence scores, means it doesn't looks that object-like. So we can ask the question, how does the neuron-guided evolution process change the objectness of images? You can imagine in these illustrations, the X and Y axis show the image plane or image manifolds, and the color green shows the objectness scores on it. And this height map is the neuron's tuning manifolds. If the two align super well, then if you climb the peaks of the neuron, you're also increasing your objectness scores. However, if you are opposite, the tuning peaks are locating off the hotspots of the objectness. When climbing the mountains, you're guiding yourself to a lower objectness scores for a neuron. So we analyze the objectness for all the images sampled by the neuron as a function of the blocks. We find that the evolution generally decrease objectness of beacon images and increase that for DeepSim images, reference to their baseline. More interestingly, it has a very difference between the V4 and IT regions. We find that when the DeepSim experiments success, then it increases the image objectness, and IT neurons lead to more increase in objectness scores than the V4, which means their peaks may be more aligned with the high object-like images. In contrast, the beacon's successful evolutions will decrease the image objectness, but it also shows the same trend between the two regions. The V4s decrease objectness more, and IT decrease less, and even stay on par with the reference beacon's reference distributions. As a control, we find this result totally contingent on the success of the evolutions. For unsuccessful beacon evolutions, if the neuron doesn't successfully guide it by their tuning functions, then the objectness doesn't decrease, which means the evolution just is randomly meandering on the beacon image manifold, and it will stay as high, so which strengthens the results of the last slide. As a comparison, the CNN visual hierarchy also use very similar results. When you're going deeper, from the first layer to the last layer, you see an increase of the object in scores of the deep sim and big GANs, but difference of their reference distributions. So as a summary, the evolution decrease objectness of big GAN images and increase that of the deep sim images, but they both achieved a higher objectness scores in IT reference to before, which shows that IT's tuning landscape is more high activation domains and more aligned with the high objectness domains in deep sim and big GAN. So finally, I will give more somehow hand-wavy explanations of how should we understand the alignment of the tuning functions and the image manifolds. So first, we remind ourselves that the tuning landscape is actually a composite of two functions. One of them is the neural tuning function in the image space. The other is the mappings from the latent space to the image space. And by compositing the F and the G, you get the tuning landscape. So it's not that surprising that landscape will look differently based on the different parameterizations. This is an example of a 2D examples that you have a 2D parameterization of UV to XY and tuning functions in XY domain as F. So if your parameterization is simply a rotations of the parameter grid, if you not distort the landscape, and the Gaussian tuning bump will also look like Gaussian. If the parameterization of image manifolds is more distorted, it's like elongated in certain directions, the peaks of the neuron will also look elongated. And also, if your parameterization is this distorted grid, then you cannot even see the tuning mountains clearly in the parameterization space. Aside from the effect of parameterization, the other potential effect is the parameterized domains. So imagine this is the domain of your image manifold, and if it's constrained to this domain and doesn't pass through the high activation domains of the neurons, then you cannot reach there by optimizing in this manifold. So these are the two potential regions that explain the alignment of the V1 to IT to different image manifolds. As a summary, when we go out the hierarchy, it's increasingly easy to drive evolutions in the big GAN image space and increasingly hard to drive evolution on the deep sim space. And we find that IT has comparable success rates in optimized activations in both space, showing that the tuning peaks of the IT neurons have a gradient in both image manifolds. We also find later IT response is more aligned with the big GANs, and the initial response are more aligned with deep sim, similar to earlier visual regions, highlighting the effect of recurrent computation may have an effect in this object space. And also, the tuning peaks of higher visual regions are more aligned with higher objectness domains in both deep sim and big GAN image manifolds. So taking back to our initial picture of alignment representation between brain and machine, in this whole thesis, we have defined many similarities between the brain and the machines and the representations. So the tuning landscape of neurons in the ventral stream and some units in the CNN visual hierarchy, we find that neurons can both guide image sampling and find peaks on the tuning landscapes. And they have bell-shaped tuning curves around their peaks, and higher and wider than what people have thought based on the classical image spaces. The peaks are growing sharper if you go out the hierarchy and with more tuning axes. And going out the visual hierarchy, neurons become more aligned with object image manifolds, and their later recurrent response is responsible for that. And also, the peaks of higher visual regions are located in the more object-like domains in the image manifolds, and their tuning curves extend beyond the objects and extend to non-object-like pattern space. In the future, I think we can explore the neural more neural aligned generative models. So we have seen this trend of increasing alignment between big GANs and visual areas in V1 and IT, but we only recorded from the posterior region of IT and not anterior regions. So we can imagine maybe this trend will still go up or it has a bump in the center. So we're not sure if going up the hierarchy, there will be more preferred image spaces or image manifolds than the big GANs. And for earlier regions, they are more optimized image generating space than the deep sim space. So in some sense, each of the visual area and neurons has its own authorized painters for it. And we can also imagine we have a continuous spectrum of generative models corresponding to all visual regions in this visual hierarchy. And in the future, we are also going to trying to understand the landscape dynamics better. So we have a descriptive views of this landscape dynamics from the broad to fine, and also from more general to more specific. But the underlying principles guiding this dynamics is still not clear. And potentially understanding that, maybe using like Bayesian inference, is important to understand why the later response of IT neurons are more aligned with big GAN image manifolds than deep sim. And we also want to generalize this paradigm to our population representations. So all the previous talks are about the single neurons tuning landscapes, but there are many mountains in this landscape, not just one. And they're intersecting and interacting with each other. So we want to understand how the multiple tuning landscapes interact to construct a population representation, like a vector in the population space. And we can even control the population vectors to interpret its meaning. And this is an example we have done in last year, which shows that we can achieve population controls through the same paradigm, but basically changing our objective from optimizing one neuron to optimizing a whole population of neurons. So the image up here is the target image, which elicits the black trace in the lower plot. And this is the alignment or mean square error between the two neuronal patterns. And here, the colors from red to blue shows the progression of the evolution process. You see, as the evolution goes on, the population pattern will change smoothly from the original patterns and gradually converge to something much more similar to the black trace, with decreasing mean square error between the two vectors. And you also see the images somehow starting to looking at the monkey face, although it's a dog or cat-like creature, but it has the two eyes and the mouth in the center of the image, consistently occurring. And this is also another image space we are exploring, which is Stelgan 2. And if you use we can also use multiple populations, like from ITs and V1 and V4. And you see that by the evolution process, the population patterns guiding the images to be more and more similar to the target patterns. And yeah, that is a brief view of the future directions of my research. And thanks for the Ponce Lab and all the members in it. And especially thanks to Carlos, advisor, and Olivia, Mary, and Liz, who help us handle monkeys and collect many part of the data in my talk. And the other members also contributed substantially to this intellectual journey. And we thank Tim Holly, who inspired many parts of this project, and grants, the Chuan grants and the CCISN Pathway Fellowships. We thank the compute centers in WashU and Harvard, and also our monkeys, Alpha and Bettles, to their contribution to science. And I'm really glad to hear that Bettel is going to retire and enjoy his life in the future. Yeah. And yeah, and these are the related publications. And thanks for your interest. Maybe. Yeah.
Okay. That was a beautiful talk, and it's really great to see all the great research you've done. And also, you know, your future directions look exciting, and you've anticipated many interesting questions in future directions. So I have some questions about those. So the dynamics part, and the temporal evolution. Have you tried to evolve based on early versus late windows of the IT response? And, you know, some hypotheses may be that the early one may be more actually like the early visual areas, while the late one may have more object-like aspects. So could you comment? That's the first one.
Yeah. So that's a definitely very good question. So based on the results or the analysis we presented here, I think it's very reasonable hypothesis that like the early, if you evolve just from the transient early response of the IT regions, you achieve closer to like the V4 representation, and it evolve from a later time bins or even later than 200 milliseconds to achieve something different. And our lab have not quite tried that. Our neighboring labs, one technician or one student in their labs have tried it in the March Livingston labs but didn't this results. So like I think this kind of motivates this to investigate it further. Yeah,
it's really interesting. Thanks, Ming-Shu. And then my second question, I'm going to take a sort of faculty privileges and ask a crazy one. So if you were doing these kind of continuous evolutions in different from neural responses in different brain areas, one sort of particularly crazy hypothesis, if you used an IT neuron, the monkey, and you just did based on IT over many, many, many days, the monkey can eventually infer the upcoming images, while in V1, if you did that, it may not. So that's my crazy faculty question. Forgive me. I'd like to hear a comment on this. Thanks.
Yeah, so that's very interesting question. Like one thing is, I think neurons, monkey are not neurons and neurons are not monkeys. So like we basically choose which neurons to evolve from, and kind of that's prototypes or that image is corresponding to that neurons. And our monkeys, hopefully they don't know that. They don't see our choice. So they don't, so I don't think from that regard, it's quite possible like explanations. And yeah, because monkey don't quite know what to expect, and different days when you choose different things. Yes. And then, yes. And if that's the case, we actually have that data. And so these are some examples from like, this is in the Octobers of 2018, 19th. And you evolve the same IT neurons, and you get this shaded yellow patterns from here and to here. So it's quite similar across the runs. And in another IT site from like September's of 19th to June's of 2020, so it's like half a year, the same neuronal sites will evolve this red eye type patterns and shows like the relatively stable across. And also this one, which across like a month or so, 2 months, if you keep evolving this black thing with protrusions on it, so which is also quite consistent. So we kind of expect it's more a property of these neurons, but if you do it longer, neuron or the monkeys may know it. Yeah.
So like how consistent are the tuning landscape of the neurons across time, but also have you compared that like the different brain state, like that they change the tuning landscape of the neurons, and what that they mean anything to the neuron or the animal?
Yeah. So by brain state, you mean like,
for example, different arousal states?
I see. Yeah, we haven't how to say that? We haven't specifically done that, but I think our approach kind of provides the tools to do it. So by searching for the peaks and finding the landscape around the peaks, then you know what is the preferred object image space to use for these neurons. And then you can track the tunings on it with different brain states. And maybe after the experiments, like the monkey becomes more sleepy or less aroused, and then you see the fire rates on that region will change. But we haven't specifically tested this like global brain state effects on it. Yeah. Thanks.
Okay, so there's no other questions. Thank you, Binshu, for a great presentation. And
actually, there's some questions in the chat. So should we?
Yeah, if you don't mind.
Yeah. So there's one from Feng Yang, and yeah, so he says like alpha and beta seem to have pretty different properties, especially with beta and have beta having clear trend from V1 to ITs, convergence time and plateaus. And would there be a behavioral difference between the two monkeys? And so for that question, we acknowledge that there is a monkey difference. So in one monkey, the V form looks more like V1, the other looks more like IT, something like that. So if you look at the trends of the second part of the talk, so in monkey A, like the V4's evolution trajectory converge more like the V1 type. In the other case, you converge more like the IT type. So we are not super sure what causes that. And we can, maybe it's like the recordings regions or the arrays regions in V4 are different. And or there could be a behavior correlates of this effect, but we currently didn't hypothesize that yet. And for meta parameters for neuron areas or monkeys, so that's also a very good question. We tune parameters for different generators or different image manifolds. Like, so one parameter to tune is the step size of the optimizers. If it has a step size comparable to the tuning peaks, then you can climb it faster and easier. If it's non-comparable, then it's hard. So we tune it for the two image spaces and keep it basically across the brain regions and the monkeys. Yeah. Yeah, Feng Yang says thanks.
Okay. No other questions on Zoom?
Not yet.
All right. Well, thank you, Binshu.
I have a question. Sorry, I'm in the Zoom. I
And,
raised my hand.
after the closed session, depending on the outcome, we may have a celebration or a morning. Either way, there will be food. This is in the Federal Learning Center in room 209.